Every cell in your body is running on a power grid that is slowly dying. Not from disease, not from age alone, from something far more controllable and far more correctable than most doctors will ever tell you. Inside each of your cells, there are anywhere from a few hundred to over 2,000 microscopic structures that generate virtually all the energy keeping you alive. These are your mitochondria. And right now, in this moment, theirs is a number that is either growing or shrinking. By 2019, a research team at the University of Colorado had confirmed something extraordinary: a specific combination of compounds found in a drink simple enough to prepare in under 3 minutes, triggered a measurable, documented, scientifically verified doubling of mitochondrial density in just 8 weeks. This isn't a supplement protocol. This isn't a clinical intervention. This is a morning drink. And what it does to the inside of your cells will change the way you understand energy, aging, and human performance forever. Part 1. The engine you never see. Your mitochondria were not always yours. Approximately 145 billion years ago, a primitive bacterium was absorbed by an early ancestral cell. Instead of being destroyed, it survived. It formed a partnership. Over millions of generations, it became permanently embedded in the architecture of every complex cell on Earth. You carry the descendants of that ancient bacterium inside you right now. Trillions of them. The mitochondrion operates by a process called oxidative phosphorylation, a chain reaction so precise, so efficient, that it produces roughly 36 molecules of ATP from a single molecule of glucose. ATP is the currency your body runs on. Muscle contractions, nerve firing, organ function, cellular repair, all of it is paid for in ATP. When your mitochondria are abundant and healthy, your cells generate energy efficiently. Your muscles recover fast. Your brain runs clean. Your immune system moves quickly. When mitochondrial function declines, something more insidious happens. Cells start operating under low energy. Inflammation increases. Oxidative damage accumulates. Fatigue that sleep cannot fix sets in. Cognitive decline begins at the cellular level, years before it surfaces at the cognitive level. The number of mitochondria you have is not fixed. This is the critical fact most people never learn. The human body has a built-in biological mechanism for producing new mitochondria. It is called mitochondrial biogenesis, and it is activated by specific molecular signals. Hey! Signals that can be triggered deliberately by what you consume. Stop. Before we go further, you need to understand something that changes the entire context of what you're about to hear. In 1988, a Japanese researcher named Yoshinori Osumi discovered a cellular mechanism that had been hidden in plain sight for decades. He called it autophagy, from the Greek self-eating. Autophagy is the process by which your cells break down and recycle damaged components, including damaged mitochondria. In 2016, Osumi was awarded the Nobel Prize in Physiology or Medicine for this discovery. Here's why that matters. The drink you're about to learn isn't just growing new mitochondria. It's simultaneously clearing out the old damaged ones, making room, and making the new ones more effective. You are not simply adding power to your cells. You are replacing failing infrastructure with functional infrastructure. Now, back to the science. Part 2, the signal that starts everything. The master switch for mitochondrial biogenesis has a name: PGC1 alpha, peroxisome proliferator activated receptor gamma coactivator 1 alpha. In 2003, Dr. Bruce Spiegelman at Harvard Medical School published research establishing PGC1 alpha as the primary transcriptional coactivator responsible for triggering the creation of new mitochondria. His team identified that when PGC1 alpha is activated, it sets off a cascade of gene expression that results in measurably increased mitochondrial numbers within cells. When you activate PGC1 alpha, you activate biogenesis. This molecule responds to cold. It responds to exercise, specifically endurance training, and it responds to certain polyphenols and bioactive compounds that are consumed orally. In 2020, a landmark meta-analysis published in the journal Nutrients reviewed 27 separate studies on dietary compounds and mitochondrial biogenesis. Across all 27 studies, 3 compounds consistently appeared as primary activators of PGC1 alpha: epigallocatechin galate, known as EGCG, quercetin, and resveratrol. Every single one of these three compounds is orally bioavailable. Every single one of them reaches the bloodstream within 30 to 90 minutes of consumption, and two of them exist in extraordinarily high concentrations in a single source. The base of this drink is green tea, not bottled green tea, not flavored green tea extract. Specifically matcha, powdered whole-leaf Japanese green tea, shade-grown for the final 3 weeks before harvest. The shade-growing process forces the tea plant to produce significantly more chlorophyll and amino acids as a protective response to reduced sunlight. This stress response is the same biological mechanism that concentrates EGCG in the leaf to levels that are, by lab measurement, between 137 and 147 times higher per gram than standard brewed green tea. A 2021 study from Tufts University measured EGCG content across 10 commercially available matcha brands. The average EGCG concentration was 221 milligrams per gram. A standard brewed green tea contains approximately 1.5 milligrams per gram. This is not a marginal difference, it is a categorical one. The second component of the drink is quercetin. Quercetin is a flavonoid found in high concentrations in a specific set of plants. Red onion carries the highest quercetin concentration of any commonly consumed food. Between 286 and 486 milligrams per kilogram of fresh weight. Capers, the flower buds of Capparis spinosa, contain approximately 1,800 milligrams per kilogram dry weight, making them the single most concentrated natural source of quercetin on Earth. A teaspoon of capers added to the morning drink delivers between 12 and 15 milligrams of quercetin. Elderflower concentrate, derived from Sambucus nigra, has been used in European traditional medicine since the early medieval period and contains quercetin alongside a unique synergistic compound, rutin, which increases quercetin bioavailability by approximately 35%. Buckwheat, technically a seed, not a grain, contains 36 milligrams of quercetin per 100 grams, which is substantially more than most fruits and vegetables marketed as quercetin sources. The third component targets resveratrol. Resveratrol is a stilbene compound that functions as a phytoalexin, a chemical defense mechanism produced by plants under stress, particularly in response to fungal attack and UV damage. It was first isolated from the skin of Vitis vinifera, the wine grape, by Michio Takaoka in 1939. In 2003, Dr. David Sinclair at Harvard identified resveratrol as an activator of sirtuins, specifically SIRT1, a protein closely linked to both PGC1 alpha activation and cellular longevity mechanisms. The most bioavailable oral source of resveratrol currently documented in the literature is not wine. It is Japanese knotweed, Reinutria japonica, a plant whose root extract contains up to 450 milligrams of resveratrol per gram, compared to approximately 0.07 milligrams per gram in red wine. Japanese knotweed extract is commercially available as a standardized supplement. At a dose of 150 to 250 milligrams of the extract, consumed with fat, specifically with the matcha, which is prepared with whole milk or coconut milk for this exact reason, resveratrol bioavailability increases between 2.4 and 3.7 times compared to aqueous consumption. Here is something that none of the studies mention. Mitochondria are not evenly distributed throughout the body. Neurons, brain cells, contain more mitochondria per cell than almost any other tissue in the body. A single active neuron can house up to 2,000 individual mitochondria. Cardiac muscle cells are second. This means that the conversation about mitochondrial health is not merely a conversation about physical energy or athletic performance. It is a conversation about the biological substrate of consciousness, memory, and emotional stability. A 2018 paper in the journal PNAS, authored by a team at the Columbia University Irving Medical Center, demonstrated for the first time that psychosocial stress directly damages mitochondrial DNA in blood cells. The paper was titled, "Mitochondria as Potential Targets and Initiators of the Stress Response." Stress does not just feel bad. It physically dismantles the architecture of your cellular energy system at the level of the genome. The drink you are building is not a performance tool. It is structural maintenance for your brain. Part 4, the preparation protocol. The drink, as validated by the Colorado research team, uses the following specific proportions and preparation sequence. 1 teaspoon of ceremonial-grade matcha. Japanese origin, shade-grown, not culinary grade. The distinction matters. Culinary matcha is harvested differently and contains up to 60% less EGCG by mass. Preparation temperature 70 to 75 degrees Celsius, not boiling. This is non-negotiable. At 100 degrees Celsius, EGCG undergoes thermal degradation. A 2016 study in the journal Food Chemistry demonstrated a 50.2% reduction in EGCG bioactivity when green tea is prepared at boiling temperature versus 70 degrees. The matcha is whisked into 60 milliliters of water, no more, using a bamboo chasen in the traditional Japanese W motion. 40 strokes, minimum. This creates a microfoam suspension that maximizes surface area and bioavailability of the catechins. To this matcha concentrate, add 150 milliliters of full-fat coconut milk, heated to 65 degrees. The quercetin source, either 1 teaspoon of elderflower concentrate, 10 to 15 capers macerated and strained, or a standardized quercetin supplement at 250 milligrams. The resveratrol component, 150 to 200 milligrams of Japanese knotweed root extract. 1/4 teaspoon of black pepper. The black pepper is not for flavor. Black pepper contains piperine, the compound responsible for its pungency, at approximately 5 to 9% by dry weight. In 1998, a clinical trial published in Planta Medica demonstrated that piperine increases the bioavailability of curcumin by 2,000%. The same mechanism applies to resveratrol. A separate 2014 study confirmed a 1.9-fold increase in resveratrol plasma concentration when consumed with piperine. The fat in the coconut milk serves a parallel function. Resveratrol is lipophilic, fat-soluble. Without a fat carrier, a significant portion passes through the gastrointestinal tract unabsorbed. The 60 milliliters of coconut milk, which contains approximately 7 grams of saturated fat, primarily lauric acid and capric acid, provides the precise lipid environment for optimal absorption. EGCG absorption is independently enhanced by the milk's casein proteins, which have been shown to complex with catechins and extend their residence time in the small intestine by an average of 28 minutes, the window in which primary absorption occurs. Consume this drink in the morning. After a minimum of 12 hours of fasting, the fasting state is not incidental. When glycogen stores are depleted after the overnight fast, AMPK, adenosine monophosphate activated protein kinase, is activated at measurably higher levels. AMPK is the cellular energy sensor that directly phosphorylates and activates PGC1 alpha. A fed state suppresses AMPK. A fasted state amplifies every mitochondrial signal this drink is engineered to deliver. Part 5. 8 weeks. The Colorado study ran for 56 days. Participants consumed the drink each morning within 30 minutes of waking, after a 12-hour overnight fast. No other dietary interventions were made. No changes to exercise protocols. No supplement additions. At week 4, mitochondrial density measurements showed an average increase of 38% across muscle biopsy samples. At week 8, the increase was 104%, a doubling. More specifically, the team measured PGC1 alpha gene expression, COX4 protein levels, a standard marker of mitochondrial content, and ATP production rates via high-resolution respirometry. All 3 markers showed consistent, statistically significant elevation. The VO2 max measurements in participants, a direct proxy for mitochondrial oxygen utilization, increased by an average of 11.4% without a single change to training volume. 11.4% in 8 weeks from a morning drink. By week 8, participants also reported secondary effects that were not part of the primary hypothesis. A measurable reduction in morning cortisol, the stress hormone that, when chronically elevated, suppresses mitochondrial biogenesis through glucocorticoid receptor signaling. A 19% improvement in cognitive task performance on standardized working memory assessments. And something that does not appear in the data tables, but appeared consistently in participant self-reports, they described waking up differently. Not energized in the way that caffeine creates energy, restless, elevated, temporary. Something deeper, a clarity, a readiness, the feeling of a body running on full power. This is what mitochondrial abundance feels like, not a symptom, a state, one final number. 1. In 1962, mitochondrial biologist Margit Nass and her mentor Sylvan Nass discovered that mitochondria contain their own DNA, independent of the cell's nuclear genome. They named it mtDNA. Every mitochondrion carries 37 genes. All 37 encode components essential to energy production. Over a lifetime, mtDNA accumulates mutations at a rate approximately 10 times higher than nuclear DNA, because mitochondria sit at the center of oxidative processes and lack the robust repair mechanisms of the nucleus. This is one biological mechanism of aging. But here's what the literature has confirmed in the decades since 2015. Mitochondrial biogenesis does not just increase the number of mitochondria, it dilutes the proportion of mutant mtDNA within the cell. New mitochondria carry clean, unmutated genetic information. When you double your mitochondrial density, you are not just doubling your energy production, you are measurably, molecularly, reversing one of the core mechanisms by which human cells age. The drink you prepare tomorrow morning does not just power your day. It is rewriting the biology of your cellular machinery. One morning, one cup, 1 week at a time. 8 weeks from now, you will not look different in the mirror. But inside every cell of your body, something ancient and extraordinary will have doubled. And everything you do, every thought, every movement, every moment, will run on more power than it did today.